Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show. Today, bringing you another really fascinating guest who is helping to create a better tomorrow on many unique fronts. Today, we have the honor of being joined by Dr. Jessica Whited. Assistant professor, stem cell and regenerative biology at Harvard University, where her lab is focusing on limb regeneration studies in axolotl salamanders, and where they're developing tools to help manipulate gene expression during limb regeneration, and ultimately exploring these various signaling events following wound healing that might ultimately initiate complex regenerative processes. Dr. Whited earned her bachelor's in philosophy and her BS in biologic sciences from the University of Missouri. She did her PhD in biology at MIT, where she studied in the laboratory of Dr. Paul Garrity, and her thesis was focused on the molecular mechanisms, in that case, controlling the development and maintenance of cellular architectures in Drosophila nervous systems. And it was during that work that Dr. Whited became interested in processes that ultimately may be required long after development events that help ensure that cells maintain their behavior, don't revert to immature behaviors, as well as understanding various processes that promote such events in response to injury. Dr. Whited worked also in the laboratory of Dr. Cliff Tabin at Harvard Medical School Department of Genetics as a postdoc studying the total limb regeneration in axolotl salamanders. And it was during that time she developed various molecular tools used to interrogate the regenerated axolotl limbs. And this is one of the core focuses of her laboratory today. She is also, in addition to all her research responsibilities, she's the co-founder of a company, Mattis Biosciences, which is focusing on leveraging many of these regenerative biology insights for next generation skincare and scar products. A lot of really interesting things to discuss today. But Dr. Jessica Whited, thank you so much for taking the time to come on the show.
Hi, well, thanks for having me. I'm glad to be here, and thanks for that really nice introduction.
Yeah, I really, I'm excited to have you here today. It's a fascinating background. Yeah, I'd love to start off like we typically do by handing the floor to you for a few minutes, just to talk a little bit more about your background. I pulled up your dissertation entitled Genetic Analysis of Maintenance of Neuronal Morphology in Drosophila melanogaster. Fascinating 500 pages or so. But I'd love to hear sort of the early days and how you got interested, sort of, not just in a lot of the molecular stuff that we hear about today, but really the study of one of the most fascinating areas of biology, in my opinion, form, morphology, sort of these complex structures that ultimately result from a lot of what's going on at the cellular level. Take us through a little bit of that early journey, if you would.
Right. So neither of my parents are scientists. But my mom was really always into getting us out in nature and those kinds of things when I was a little kid. And so we spent a lot of time camping and hiking and things like that. And I grew up first half in Michigan and second half in Missouri. And so most of our fun time was outside. And I remember we collected butterflies. I don't do that anymore. We just take pictures now, but we were really into collecting butterflies, and I had a big rock collection. And my sister and I spent most of several summers in a creek near the house, like catching crayfish and these kinds of things. And I was always just really fascinated by sort of like critters and what they're doing all around us, and always finding things like that to play with. And so part of me always knew that I might go into something scientific, but I was also really honestly interested in politics, which is hilarious now because I could never see myself doing that. And so when I went off to undergrad at the University of Missouri, I was, really just sort of poking around, a little bit clueless about what happens after you get a bachelor's degree. But I had gotten interested in philosophy. And I knew that reading philosophy texts and thinking about philosophy, I was like really where my heart was. I loved it so much. I loved taking the classes, all of it. But in parallel, I got to school and I needed a job. So I got a work study job, which in the US is like a federal program for helping students basically make some money while you're an undergrad. And I had to pick a place to do my work study assignment. And I remember going in and there was a big 3-ring binder. So this is pre-computer situation. Computers existed, but we weren't using them for all the things we use now. And there was a big 3-ring binder and I was going through it and something really captured me. And given my background, it might not surprise you that this opportunity was in a soil science laboratory. So at the agriculture school, which of course is really big at the University of Missouri. But the soil science laboratory was basically studying the species of earthworms that lived in different types of soil in Missouri and sort of relating them to soil quality and et cetera, and these kinds of things. So I thought, well, if I'm going to have to get a job anyway, I would rather do that than to go and like work at the library desk or, something that I saw as more mundane, right? So I went there and it really kicked my butt, right? This is my first experience working in a professional laboratory setting, and there were not a lot of other undergrads there. I think I was the only one actually. And I didn't know anything about, graduate school, what people go to graduate school for, postdocs, and like what the professor really does and these kinds of things. But it was like the Wizard of Oz or something, like where at the end where they sort of pull the curtain back and you see, and that was the first time I really saw behind the scenes, like getting to pull the curtain back. And there were many other times later. So I spent 3 years there, working part-time during the school year and then a couple of times full-time in the summer, and it was really hard work. But it taught me a lot of discipline, which came in handy later because the stuff I went on to study later in my career is just so freaking hard that I think I needed like to see, to build that foundation, right? And so the kinds of things I was doing there was taking chemical inventories, cleaning glassware, literally sometimes I would spend 4 hours cleaning glassware. I would leave soaking wet. And that's how I got my start in science. And then I worked my way up. And so initially I wasn't doing anything with the worms. I was sort of lusting after them. And then later I was allowed to do like very specific things with the worms, to try to figure out what species were in which kind of soil and these kinds of things. And then eventually it got a little bit more sophisticated whereby I would have to dissect the worms to distinguish the species. And that was really what was starting to get me going, right? But, I also wasn't in it for, in order to learn about agricultural practices or to like go into agriculture, right? And so I knew that probably that wasn't my ultimate home at the university. And so at the remainder of my time at Mizzou is what we call it, I went into the traditional biology department, which was more like in arts and sciences, right? And I joined a lab there doing something totally different for the last year. And my advisor there really sort of schooled me on about what graduate school and what it is. And like, given that she knew me pretty well, what kinds of things I might be interested in graduate school. And that's when my radar went off and I was like, okay, so maybe I should do this instead of going to graduate school in philosophy, which is really what I was thinking. The other thing that happened is that my philosophy advisor, his name was Alexander von Schoenborn, and he, was like the kind of guy that would wear tweed and he had a pipe in his office. And I've done really well in philosophy, but he was super straight with me. He was just like, Jessica, I would give my left big toe to be a biologist in this era. And mind you, this was about 1998, 99. So even then he was saying that, and it was, really like he had a crystal ball or something, because it turns out that biology is, the next several decades in biology were just killer, right? And they still are. And so the fact that my own advisor was sort of steering me away from going to grad school in philosophy for various reasons we don't have to get into, that's basically when I made my decision. And so I really thought so I did like this huge interview circuit, which is typical of people. And I really thought I was going to go to one particular school because it sort of seemed like my fantasy school. I remember being so excited to get the interview. But then as I did all the interviews, I realized that there are other things that are important to me other than, what I thought were the things to look for initially. And one of them was cultural fit. Like, how do you see yourself in that program with those groups of students, et cetera? And importantly, who do you see yourself working with when you go off? And I had met this guy, Paul Garrity, at MIT during my interview, and he just totally captivated me with the topics that they were working on. And it was not center of the plate what I thought I would be studying at first in graduate school. But after I met him, I felt like, well, this is probably what I want to do, right? And so I ended up going to MIT and I did work in his lab. It was a fantastic place for me. I learned so much working there and it was extremely intense. And yeah, I got back to insects, which is kind of where my biology interest stemmed from. Of course, I wasn't like studying insects in the wild. I was using this model system, which we can talk about. But that's basically how I got to becoming a professional scientist some years ago. Yeah.
Outstanding. Yeah, it's fascinating when I hear obviously guests talk about nature, the reverence for nature. You're coming right now just in recent days, and we were chatting before the show about sort of setting this show up at the opportune time. But the amazing sort of this two-part editorial that you just recently published, the American Association of Anatomy, Journal of Developmental Dynamics, just about a week ago or so, entitled Salamander Models for Elucidating Mechanisms of Developmental Biology, Evolution, and Regeneration. And I'm a huge fan of I refer to them as sort of all the species with the really cool superpowers. So, I love extremophiles, I love the immortal jellyfish, planarians, and so forth. But axolotls have that unique space in my heart. Well known for this amazing regenerative capability, limbs, spinal cords, eyeballs, parts of the brain, all that stuff. But then, as I'm preparing to talk to you today, I want to make sure I knew enough about things. So interesting facts I learned in preparing for the discussion. The only species of amphibian that does not undergo metamorphosis, I know that. 32 billion base pairs in the genome. 10 times the size of the human genome. You have been extremely influential in establishing a breeding colony of this species, which is very difficult to find in the wild anymore. So introduce us a little bit more to the axolotl, because it really is, amongst the superpower species, as I like to refer to them, one that we're going to be learning a lot, not just in 2022, but as you were saying, the decades to come, we're going to be learning a lot from them. Talk a little bit about axolotl, if you would.
Right. Okay. The first thing that's like really obvious about them is everyone can appreciate they're actually super cute.
Yeah.
And they're really popular right now in popular culture, and they were before Minecraft introduced axolotl. But those of us who have kids are probably well versed in this. And so I get a lot of kids now writing about that, but mostly they're writing about the science, right? And so, if we back the truck up a little bit, when I was working in fly neurobiology, I thought that stuff was just so interesting and I loved it so much and it's so important and all these things. And one thing I really struggled with though is explaining why I care about it so much to a lay audience, to relatives, right? And then when I struggled with that, I realized that actually what I want to do with the rest of my career is something where I can just say it in one sentence and a 7-year-old will appreciate it. And the way I like to think of this is that wouldn't it be awesome? Everyone agrees it would be awesome if humans could regenerate limbs because there's this huge clinical need, and a real limb is always, by definition going to be better than anything that could be engineered, right? So that sounds like science fiction, and I would say it surely is, unless we know we have an instruction manual on where are you going to get the instructions. And it's nature has provided these instructions to us. We just have to crack them. Right? Because salamanders, basically all the salamanders that have been studied to date, they all regenerate limbs. And people have known about this for several hundred years in the scientific literature, but maybe more even before that, right? And it's just been a really difficult scientific problem to crack, to understand like where are the cells coming from and where are the instructions that they're receiving coming from, and how do those cells undergo the changes that are required to make a new limb. And the reasons why it's been harder to crack than some of the other mysteries in biology are ones you just elucidated, you sort of talked about, which are articulated, which are the genome is super huge. And we're going to get to the axolotl in a second, but the genome is super huge and there weren't a lot of genetic tools. And as modern biologists, what we really want to say is we want to say, well, what do all of the genes and cells do in an organism and a process we care about? The gold standard for asking those kinds of questions is to say, okay, I care about this process X, and I think gene Y is involved in it. So let's get rid of gene Y and then see what the effect is. And then you work backwards and you say, okay, now we know that gene Y is required for this, right? And it was just basically almost, you couldn't do that in salamanders until recently. So why the axolotl amongst all the salamanders? Because actually it's not like the smallest one or anything, and that's an important consideration if you're going to grow a whole bunch of them in a lab, especially a lab in the city like here. It's not the smallest, but it has the shortest generation time of any of the easily obtainable ones. And when I tell you that it's 1 year, it is a little bit scary. Still, it takes 1 year to go from an egg to a reproducing adult. About. Okay. A fruit fly, for reference, is 2 weeks, and a mouse is something like 6 weeks. Mm-hmm. And so it still takes a lot longer, which means you have to invest a lot more resources into making these genetically engineered animals, for example. Takes several years to get enough that have the same mutation for you to do that study that I just told you about, right? But that is, I think, the primary reason why people who are studying molecular biology of limb regeneration have turned to axolotl versus some of the other salamanders. Mm-hmm. And then, there are some other types. There's a particular kind of newt that actually has also a generation time that's on the manageable level. But these are basically the two primary models for those reasons. If you look in the older literature, there are other kinds of newts that people frequently use, but if you dig into the methods, those are mostly collected by some dude named Charles in Tennessee. And he literally used to get them in the wild and then give them to labs, or sell them to labs. And they were not being actively bred in labs. And so if you want to do real genetics, you have to have a handle on the life cycle from start to finish, and you have to be able to breed the children and the grandchildren, et cetera, of these salamanders. So this is why we're using axolotls. And as you mentioned, the genome is ginormous, so it's actually 10 times bigger than the human genome. And luckily, one of my colleagues has sequenced it. But you might also, so this is Ellie Tanaka, and her colleagues have sequenced it a couple of years ago. Mm-hmm. But you probably also know that when people sequence genomes, the first draft is never, any genome is going to have some parts that are not 100% sequenced, unless you're talking about something like an E. coli probably or something, right? So we do have this draft, and it has been super awesome to have. But it doesn't have every single iota of information in it. So there's that now. And if we back it up a little bit before that publication, what we were doing, and other people too, was making these things called de novo transcriptomes, which is basically something where you catalog all the genes that are expressed in tissues that you care about. And then you take them by sequencing the RNA, which is the product of the gene before it goes to actually make protein. So it goes, DNA, RNA to protein, right? In most cases, right? And so in this case, what you do is you say, I want to know all the genes that are expressed in this regenerating limb. So you just grind it up, you purify all the mRNA, and then someone has to assemble it. And we have been super fortunate to have like a guru of this kind of business collaborating with us for a really long time. And his name is Dr. Brian Haas at the Broad Institute here in Cambridge. And together with Brian, we made a de novo transcriptome several years ago for the axolotl, which was really catalytic for our lab's work. And I know a lot of other people use it too, but it really was just instrumental for us to get in there and figure out okay, well, what genes are even active and which genes are repressed, and can we make hypotheses from this information? So yeah, that's basically what the thesis of the lab is. If we understand how this is happening in salamanders, then other people can take these insights and use them to try to augment regeneration in mammals in the future, including humans. And so I really think people ask me, when is it going to regenerate? When are we going to regenerate human limbs? And well, of course, I don't know the answer to that, but I always say it's a function of how many people and how many resources, because I think as humans, we will get there. but it's, you got to learn how it happens. So
absolutely. Yes. Absolutely. And, drilling down on that a bit, because, you approach the limb regeneration problem from a variety of angles. And I think the core place I'd like to start off, as you're talking about some of these tools, and a core part of the axolotl, but also some of these other amphibians, regenerative armamentarium is this really unique cellular structure known as a blastema, where all sorts of cellular and other tissue types of magic are occurring. Cells are de-differentiating and redifferentiating. You have all sorts of extracellular matrix that's being broken down and built up again. Gradients are being, a lot of things happening in the blastema. It's sort of like, I compare it, obviously, thinking about it as sort of a whole new embryo that's beginning and going to create that new limb where the limb was lost. As you go about, I mentioned in the beginning that, a core of some of these tools that you've been developing is to interrogate the blastema. How do you start interrogating the blastema? You got these amazing tools, you got a lot of genes and stuff, but there's just so much happening there, multiple cell types. You say all this other stuff that's happening, and we'll get into some of the grander topics later in terms of positional identity and all that stuff. How do you start? Like, where do you start in that blastema, which is such its own little universe of regeneration?
Right. Okay. So we will connect this to the tools. So in the old days, people, and including my postdoc advisor, Cliff, they would say, okay, we've learned as a field so much about how limbs develop. Probably it makes sense to hypothesize that some of those same genes are used during limb regeneration. So a lot of the early work was basically asking those questions like, okay, somebody else identified this gene, let's say sonic hedgehog, right, which is a gene that Cliff is really well known for. And if you express extra sonic hedgehog in a chicken or a mouse, you can make extra digits. And my colleague Stefan Roy in the lab of David Gardner showed that if you express extra sonic hedgehog in regenerating axolotl limbs, you also make extra digits. And if you block it, you reduce the number of digits. So there are examples like this, and they were mostly inspired by the knowledge that had already been built and the genes that had already been identified in limb development. And it turns out that once you make a blastema, many of the rules look like they probably apply, insofar as it is like a giant limb bud, which is a structure which is similar to a blastema, except for the embryo grows it, or in the case of the salamander, a hatchling grows it, and then it leads to the new limb, right? But those approaches are awesome, but they're also kind of not completely satisfying, because you could also postulate very reasonably that there might be genes that are either completely unique and required for limb regeneration, but not limb development, or they're used in a different way, but they're both doing something, right? And so you really want a more agnostic approach to asking those questions, right? And when I think about agnostic approaches to discovering what genes do, there's really two ways, and this comes back from like MIT biology days, right? So there's something that's called forward genetics and reverse genetics. And forward genetics is the much more sort of old school thing, whereby someone does what's called a screen, and they say, hey, I want to know how these antennae get made in this animal. And so they mutate the crap out of the animal, a whole bunch of different instances of the animal, and they tweak it so that, there's like, a lot of detail that I'm going to skip over here. But they basically say, okay, now we have these 1,000 animals, and every single one of them has a mutation in one discrete separate gene. Which ones don't grow antenna, right? And now we're going to work backwards and say, of the ones that have this mutant phenotype we care about, which is they don't have antenna, which means the gene might be required to grow it. Like, let's map this gene and then figure out what it does. And that is still something that we are working towards actually right now in the lab. But when you're talking about a 1-year generation time and an enormous genome and all this stuff, it is much more complicated. You have to be a little bit more nuanced in the design of these things. So that is forward genetics, and it's glorious, right? You're going to find stuff you wouldn't have predicted if you do that. On the other hand is reverse genetics, and the approach I described to you earlier about using clues from limb development. That's one example, right? Like, basically somebody is serving you the candidate list from these other studies, and now you're going to go see what happens, right? And our early days were also reverse genetics. And working with Brian Haas, we made this catalog of all the genes that, let's say, are up-regulated in the blastema, but they're not very highly expressed before the injury, right? And I like to think of this as like, if you were to go to another planet and you met some aliens there and they have some other organ that you don't have, and you're like, well, what the heck does it do? You know, you might want to basically just say like, what factors is it making, right?
Yeah.
And maybe learn something that way. So that's what we did. We said, what factors is the blastema making a lot of that are really not very highly expressed in uninjured limbs? And then once we know their identity, and then we go back, so identity computationally, then we go back and validate them, which is an experimental enterprise. Then we go and say, if we mess with their expression, do we break limb regeneration? Do we stop it? Do we slow it down? Do we make it look weird? And those kinds of things. And we did that for many years in the lab, and we're still doing it. But, you know, at the same time, it is subject to the caveat, which is like, you can go in and could have all the evidence in the world that this thing might be really important, but it could turn out to literally just be a marker. It's like a gene that the blastema is making, but it's dispensable, right? And we've gotten that result many times, and many of these are unpublished, right? Because it's sort of underwhelming, right? So to be like, okay, we got rid of this gene and that doesn't really look like it does anything. And it takes so much effort even to get to that conclusion that, you know, in the lab, we're actually trying to push the technologies to try to do the other kind of genetics, the forward genetics, in, you know, a more refined way though, here in the lab, so that we can figure out what genes drive this process. Okay, so that's the first part of your question. The second part of the question is basically, is it really true that we're going to, even if we say we're going to take a reverse genetics approach, are we really going to do this just by grinding up like these huge structures? Because as you said, there's a lot of different kinds of tissues in a huge structure, like a whole limb. And even within individual tissues, there's multiple different cell types most of the time, right? And so there's a whole sort of smorgasbord of cell types, right? And I'm not really one of these people that wants to, you know, define the cell types for the sake of knowing them, or to define like how they're related to each other. But it is important to do that in order to formulate hypotheses about who is doing what. And one way that you can do this now is to do what's called single cell RNA-seq, which is basically to dissociate all the cells from a tissue. So when I say dissociate, I mean get them free from their extracellular matrix, which is the goo that holds cells together, right? And then basically what you can do is a bunch of fancy chemistry whereby every single cell gets sequenced individually. And so you can see all the mRNAs that each individual cell is making. And then you can compare thousands of them to one another. And then you can make more granular statements about gene expression. And that can really help you figure out like what the exact cells might be doing, for example. So we've done some of that now since those original transcriptome papers, as we call them. And that's allowed us to, you know, refine our hypotheses. And many of those projects are still works in progress, but that's basically what some of the things that we're doing. And along the way, we actually discovered several phenomena that we didn't realize we were going to sort of walk into. And some of that has illuminated the process of limb regeneration. And those are some phenomena that we've been really digging into with these kinds of tools now. So, you know, we study straight limb regeneration, but we also study things like what's happening elsewhere in the body, which is the project that I'm talking about most often right now coming out of the lab.
And that's you read my mind, because that's next on my list here, because separate from the blastema, you have this focus on the systemic components of what's happening. You wrote a paper, I saw, cross-species analysis of systemic mediators of repair and complex tissue regeneration. We've read a little bit lately, last couple of years, about the immune system. And how in some of these more ancient species, you know, they don't have, you know, they have a lot more of the innate immune system than we have, a lot less adaptive, and there's also interesting interleukins and other cytokines and so forth that have pro-regenerative capabilities. What are some of the things that, obviously nothing confidential that you're still working on, but what are some of the things you're finding in terms of that systemic milieu that is affecting local regeneration?
Right. So several years ago, we did an experiment whereby we looked and saw what was happening on the limbs, which we call contralateral. So when I say contralateral, I mean, if your left arm is amputated and you're a salamander, most people are studying what's happening on that left arm, right? As a local process. But we noticed that on the other arms, that they're not innocent bystanders, right? Like stuff is happening over there, and some of the stuff that's happening over there is actually fairly overlapping with what's happening on the amputated arm. And so we were just scientifically curious, like why or how is that happening and why? But I actually think it's a clue also. It's a clue to this process of building a blastema being something that I like to think of as actually 2 steps, and the steps are separable, like conceptually. The first step is that after an injury, there's some kind of system in place whereby stem cells get activated, let's say. And when I say activated, I mean they're told to start synthesizing more DNA in anticipation of dividing, of making more of them or proliferating, right? And that is one step. But a second step is that some of these stem cells have to go on and, quote, become blastema cells. And we like to think of that in my lab as a transition to the blastema cell state. And you can imagine that both of those things have to happen at the site of injury in order to get a limb to regenerate.
Sure.
But the fact that we discovered that the first step is happening all over the body, but yet this animal doesn't grow extra legs everywhere, suggests that it's actually, elsewhere in the body, the first step is engaged, but the second step, for whatever reason, which we're still figuring out, doesn't get engaged. And this tells you something about order and sort of like how cells know what to do. And I think it relates to regenerative medicine prospects also, because what we are now doing is characterizing the full repertoire of these cells elsewhere in the body that undergo activation in response to an amputation, and basically trying to relate them to what is known about stem cells in mammalian systems, which, as we know, they don't regenerate full legs, but they can regenerate like fingertips. And how similar that is to full limb regeneration is still a fairly open question, but they don't regenerate full legs. But if they do activate the cells, then I think that's a very different thing than saying, oh, it's game over and mammals just don't do it, right? It's possible that mammals do the first step, but they can't do the second step without intervention, and that salamanders are exquisitely good at converting those activated cells from the first step into bona fide blastema cells. And so we are really been digging into those questions about what's the consequence and the mechanism of this systemic activation elsewhere. Right, so that publication, the first one, which is sort of like, oh, here's the phenomenology, that was a few years ago. And right now we are working on sort of the final experiments to really say something about the consequence and the mechanism of systemic activation, how it relates to limb regeneration.
Awesome, awesome. Yeah. Continuing along that line, I noticed you've published a few papers with Mike Levin at Tufts. He's been a guest on the show a couple of times, and we talked a little bit about positional identity and how some of this old going back 100 years or so, a lot of these regenerative laws that if I was that salamander and chop, I never really get a hand here. An arm will grow and a hand will come later. One of the ones that stood out, though, to me was this one on limb bud size, repeated removal of developing limbs. And I remember when I was chatting with Mike, this is about a year ago now, he pointed out that size control, even more than positional identity, is one of those really weird things in the sense that, we got to make sure left and right don't get mixed up. But at the end of the day, I want a hand that looks like this. I don't want a giant hand, or I don't want a really teeny little hand. And it's kind of a mystery how all that happens. What are some of the things? I mean, are you looking at some of the bioelectric stuff that he's working on? Any other interesting biophysical type studies that you're doing to really explore some of the mysteries of both position, but also size and shape?
Right. So Mike is a good old friend of mine, and yes, we have collaborated with him. And I guess I just would give the disclaimer that right now we're not, my lab isn't incredibly focused on those questions, but we have stumbled into some of them, and they are really big and important questions. I like to think of, we want to understand how to make a blastema. Of course, the rest of it's also really important, because blastemas are programmed in this way to grow back exactly what they need to grow back, and no more, no less. And so, you're right, you want the exact right form to be regenerated. But because mammals don't naturally make blastemas in response to a full limb amputation, that's why we had to kind of contract and focus specifically on that question. Now, going back to that study that you talked about, which is, by the way, of my favorite studies from the lab,
it was really cool.
limited. Yeah, it's really cool. It was limited though, because we have it in that paper, we didn't drill down and find out the mechanism. But it was really interesting, and it's doing that experiment. Actually, it stemmed from my philosophy background. And what I wanted to do, but I couldn't explain it this way in the paper because the reviewers would just hand it to you, was that what I was thinking was that, you know, in these super regenerators, they can grow a limb at any time in their life. You know, this animal could be 10 years old and suddenly it's out one day and it gets its arm bitten off. And we're going to talk about that in a second. But it gets its arm bitten off. It had no idea that morning. It was like eating breakfast. It had no idea that it was going to have to grow a leg back, right? So random. So it got me thinking, like, is it possible that these animals harbor the information required to grow the limb for the first time at these like weird late random times in their life? Like, you know, this is an interesting question, because is one of the reasons why humans can't do it, because they cannot recollect that sort of like blueprint, right? Of how to grow a limb. And if we knew if super regenerators can make their first limb even when they're an adult, that would be interesting, right? And so the way that I wanted to tackle this, and my first graduate student, who was just a fantastic person and scientist, he did this project, and it required him to remove the limb buds, which are the structures that are they're like these little balls of cells, and they give rise to the limb. And basically to just repeatedly remove the limb buds for like 9 months, and it's like a little biopsy, and then don't let them grow a limb, and then one day stop and see what happens, right? And we were both surprised at the result. I had done a little poking around when I was a postdoc, so I wasn't like flabbergasted, but I was like, this is actually really interesting. So the result he got was that was extremely binary. Either nothing grew, so in that way you could say that they're completely blocked, or they're broken, or they're exhausted, or whatever, or something grew, and it looked almost like perfect on the outside, and it was perfect on the outside, but it was miniature. And the cool thing about it being miniature was that you couldn't argue that, oh, you know, it just needs to catch up, because we let those animals be in the lab for like a couple years, and they never caught up. Right. And we didn't do this to the hind limbs, so we always had those to compare to. So they were permanently miniaturized. And I still think that's like a really interesting outcome. And, you know, we're still drilling down to figure out why we got that. But it's cool to think about a couple of things. One is that we also showed in that paper that if you reset the limb, and you do that by amputating those miniature limbs, what do you think? I mean, you might have seen the paper already. So what do you think happens if you amputate those limbs? It could be that it seemed equally plausible that if you amputate them, then they grow back a limb that is matched to the size of the body, so they reset. Or they're permanently miniature, in which case you would argue that the information about the size control might just be local, right? Because if it's permanently miniature after regeneration, that might be what you would expect. And that's what we saw. We saw that the miniature limbs were permanently miniaturized even following resetting by amputation. And so I think that's a pretty cool finding. Again, it's been hard to track down, but we're making a little bit of progress here on, you know, what is happening. But we're not really studying the limb size per se. We're actually studying some stuff that's like more related to, you know, the plasticity of those cells in the regenerating limb. In those two different contexts. So that's interesting. And then the only difference we saw besides the size in those miniature limbs is that if you get inside there and you start to look at like all the tissue types that are inside there, we saw that there was insufficient innervation. So there just weren't as many nerves in those limbs. And that really honestly didn't surprise us, because there's a huge body of literature that has already documented that nerves can actually directly control size of organs or appendages, etc. There's this really interesting human condition whereby people grow like a giant thumb or a giant index finger, but the rest of their hand looks normal, and that has been tracked to, you know, exuberant innervation, right? And so you know, we had peered in there and we saw that, but everything else looked almost basically perfect. And one of my colleagues here at UMass Boston has since followed that up and found that there are particular attributes of the nerve that might be molecularly sort of controlling this. And she's done this interesting work where it's not just like, oh, you know, they formed miniature limbs by virtue of this experiment that they did, but she's basically leveraged this thing that she calls it the tiny limb, and we call ours the mini limb. But the tiny limb basically is a phase that a regenerating limb goes through before it gets to acquire its full shape. Naturally, it happens. And so she's kind of linked it to this. So anyway, like, I like to think of that as like, what the sort of the, can the animal make the platonic form of a limb in its old age? And, you know, then I like to think about what that might mean, but we're so far from really connecting it to human regeneration. But yeah, that's a cool paper. But one thing that I wanted to get back at is this idea that the animal's losing limbs randomly, you know, through predation, for example. Because people ask me all the time, you know, why can salamanders do this and humans can't? And I got to tell you, I think all the answers are just hand waving right now. But I'm about to provide a hand waving example here. Salamanders, a lot of salamanders, especially, you know, the ones whose eggs are laid in these things called vernal pools, which are like glorified puddles of snow. Okay. Like if you go in New England in the summer, you'll in the spring, right after the snow melts, and we did this year, my boyfriend and I, you will see thousands of salamander eggs in like a very small space. And it really is sort of survival of the fittest, because their main sort of predators are other salamanders of the same species when they're in those high concentrations, because they're, you know, those pools are ephemeral, and so they're not filled with like, you know, mean fish or something, right? And they're incredibly cannibalistic, and they're incredibly cannibalistic in the lab. Even the axolotls, which apparently, you know, in the wild, they live in a much bigger lake if you can find them. But even in the lab, if you house them together, they just eat each other's arms and legs. So if you house the babies together and for any amount of time and think you're going to go back and study limb regeneration, that is, in my opinion, not a good idea, because the first thing you're going to have to do is let them all try to grow a leg. And I found out pretty early in my career that if you do that, and you take a bunch of them that have been eating arms and legs and you separate them, you know, some of them don't grow arms and legs. And that got me wondering why. And this goes back to this other paper, which is kind of connected to the one you talked about, but this other one is just repeated amputation. So instead of not letting the animal grow the limb, we basically wait until the animal's grown a limb. And then we ask, like, how many times can they actually undergo this? And we found there's a limit to it. In fact, after 5 amputations, 60% of them couldn't grow anything at all. They didn't even get a blastema, right? And so, like, going back to this idea that maybe something like cannibalism is a selective pressure that animals that are exquisitely programmed to be able to regenerate limbs actually undergo a significant amount of cannibalism in the wild. And so they really feel it, right? Like there's this not zero chance that it's going to happen, right? So kind of going back to the systemic thing, which we're working on a lot right now, one of the things that we found is that if a limb is amputated and then the stem cells elsewhere in the body get activated, that, in this activated state, they're actually poised to go on and make a new limb if they get their own injury to respond to locally faster. And so I think that's really cool, because, you know, you would think that, you know, who cares exactly how fast you do it, but if you're the salamander and you need to crawl around in order to survive, and you've already lost one limb and there's a lot of other critters around that could eat you, you know, your cousins, then it makes sense that it might actually be something that you might want to retain or build upon the molecular program to have this sort of poising happening, and get the cells ready. And, you know, we could talk for hours about some of the old literature that adds up to hinting at this being the case, but our recent studies have shown that these distant cells are actually poised to respond faster to injury because they had an amputation elsewhere in the body. And I think that's really cool. And then the second thing that I think is cool about that is that the sort of straightforward explanation for that links up to some really cool work that has been done in mammals from the lab of Tom Rando, showing that mammals have these injury responses that are systemic also, and it involves stem cells getting into like an activated state. But as we discussed, they don't regenerate full limbs. And his work showed that the signals that sort of sprinkle around the body and get the stem cells to activate are carried in the bloodstream. And so, you know, that is a totally realistic hypothesis, and one we actually haven't ruled out in the axolotl. But in parallel, we tested the role of the peripheral nervous system in activating these distant cells. And we found that it's absolutely required for the activated cells to get activated in response to a distant amputation. And so now we know that there's another way that cells can get activated, and in these highly regenerative organisms, they're using that way. And so we have done a bunch of molecular studies to try to figure out, well, what is it about the nervous system that is sort of providing the signals to do this? And thus far, we have shown that one of the things that is an input is the adrenergic system. So the listeners might know about adrenaline, also known as epinephrine.
Yep.
This is the thing that is like the fight or flight response. And it's interesting because this is one of the ones that looks like it's feeding into this activation response, which I think is really cool because, you know, if we go back to this cannibalism thing, then it's totally possible that the adrenergic system gets like sort of turned on after the injury, and that can help prime the whole body to be in this activated state. And so, yes, that's more or less what we've been sort of mulling over here in Cambridge for the last like couple of years, to tell you the truth.
No, it's a really cool. I mean, the whole systemic stuff is cool on its own, but what you're mentioning about the cannibalism and the, I mean, I think it was Bruce, a couple of years ago, I did an episode with Bruce Carlson, University of Michigan, and he talked about sort of this principle of induced phenotypic plasticity. He was talking about some stuff. I forget exactly what it was, but like if you could make the sound of a snake near the eggs or something, you make the baby frogs nervous. I forget what the whole thing was, but it kind of like you're bringing it back down. I'm like, wow, this is it. Yeah.
Actually, this is really interesting because I don't know that literature, so now I have to look it up. But I've actually never gotten to meet Bruce Carlson, but he has some of these like seminal older papers. Probably when I was a toddler, and I knew he was at the University of Michigan. And he has this one that I'll never forget. I think, he basically did something, and I swear he might have used the analogy of sausage. In the scientific paper, where he basically took the muscle out of a salamander limb and minced it up, and then stuffed it back in there, and then and waited for it to heal, and then amputated, and to see like, oh, is it going to grow back a normal limb or not? You know, and I think that the result was that it did. So in computing like how to make the new muscle, the process of regeneration, it basically corrected the problem, right? And there's some other cool papers like that involving like removing part of the skeleton or this and that. And so the amount of plasticity that these animals have is, I think, really impressive. But yeah, I would love to meet him. I'm impressed that you got him on the podcast. I didn't know that.
He was doing stuff with lakes and fish and other things, but because he retired now, it was we'll chat about that offline. But yeah, there was interesting dude, and I always hold his Principles of Regenerative Biology as like one of the Bibles that I keep on. I don't have it on this shelf, but it's like I love. Speaking
of these people, maybe you can help me get a hold of this guy Roy Tsava, because his older work, and I tried to reach out to him, but I don't really have a great way of doing it, and I don't want to seem like a stalker. But anyway, I think he's in Michigan, like retiring on a lake there. And he had some really cool old papers that really speak to this kind of systemic activation that we're talking about, even though he didn't call it that in the paper, but the poising, like the cell poising. Some of his old work, I think, really hints at that too, where they sutured the limb. And then if you suture a salamander limb, it also doesn't grow back. So if you do what we do to humans to salamanders, then they don't grow back. Right. But if you suture the limb and then you pop the sutures open, like, 2 months later, then they grow back, and you don't need to re-amputate them, which is so cool when you think like, oh, they remembered that they're injured, but also they grow back faster than they would if they got a primary amputation then. So to me, that says that maybe the cells were activated, they did step one, and then they're just like being held by the organism in this activated state, primed, right, for what's going to happen next. So I would love it if you can get a hold of Roy Tsava.
I'll put that on my list. I will put that on my my job jar. I will take care of that for you. Not an issue. But let's one other thing I want to ask you about, because, and once again, I know limb regeneration is the core focus, but I have to ask you about it because you published on this, and then once again, we're talking about papers from yesteryear. You have this paper, Parallels Between Wound Healing, Epimorphic Regeneration, Solid Tumors. I think you wrote it with Mike, actually. But, we go back, you and I 80 years in the literature, and there's all these fascinating studies about people that dumped all sorts of nasty things, cancer cells and pieces of tissue and all into blastemas, and the nasty stuff just goes away and you get the normal limb. And the tumor, and I'm sitting here in downtown Philadelphia, and down the road, sort of the human parallels for this were done in the early 70s by Beatrice Mintz and teratocarcinoma cells and embryos. The axolotl also, while it's known for its regenerative capabilities, has this amazing, what we can call it anti-cancer, whatever you want to call tumor reversion capability. I know you focused on the regeneration stuff, but any other interesting insights that are coming out of your limb regeneration work in terms of just this principle of how a microenvironment, whether it is a blastema or an embryo or whatever it may be, helps normalize bad cells or normalize and incorporate them as normal tissue? Because I think this is another really fascinating thing these species can do.
Yeah, it's so fascinating. I got to just give the disclaimer that my lab has not really done a lot of work in this matter, but I do think it's interesting. And what the paper you're talking about is a review article that I did with just really spectacular undergraduate who's now an MD PhD at Harvard, and studying cancer per se.
Awesome.
So, you know, like that literature is so interesting. And there's so many old papers. Let me just highlight one that you didn't talk about that is my favorite one to talk about in this topic for class, which is this business, Charles Bredes. It's a one author paper. I think it was from 1952. And he basically gave newts a carcinogen. The one he used was coal tar. And coal tar has a bunch of organic compounds in it that in a rat, for example, will cause a tumor if you, stick it under the skin or whatever. And what he found was that if you do this to newts, they very rarely get tumors. Like hardly ever do they get a tumor. So few tumors in the hundreds that he tried to induce the tumors in that he could show like a picture of each of them, right? And instead, what happened was that they typically regrew, they grew an ectopic limb, like an extra leg at the site where the stimulus that would make a tumor in a person or a rat was placed. It's just crazy, right? And it's also crazy to think about how it happened. Well, we don't know the answer to how it happened at the molecular and cellular level. But while it was happening, if he cut those little nascent limbs off, these extra limbs, and he went and looked inside there, he saw that it looked indistinguishable under the microscope from a bona fide blastema. So like the limb grew in the same way that it would grow following an amputation from the outside world perspective. Now, nobody's gone back and like repeated that and then taken some of these more granular molecular tools and like dissected it, but that would be a great thing for a person to do. And then if you dig into the literature a bit more, you also see some hints, and I think someone should reinvestigate this, whereby when you do occasionally see a tumor in a salamander, if you bisect the tumor in the process of amputation, it will go away, the half that you left on the body. And like you said, it will basically tame a tumor cell. And, this is really interesting, and certainly our lab can't do everything. So we're really not digging deep in here, but someone should do this, right? Which is basically to say, okay, most of the treatments for cancer are targeted at, well, the word targeted isn't even, nowadays, targeting is a huge deal, but most of the treatments in general are to kill cells, right? You kill tumor cells. But what if you could instead retrain them, or instead sort of like instruct them to go do something that's not nefarious, right? Like, why don't you turn into a muscle cell instead? And then the person, isn't going to undergo metastasis and may not, have a really bad outcome from this tumor, right? So I think there are things to be learned by going back and doing the types of experiments you just talked about, but with a modern lens for sure. That would be interesting. In our lab, one of the things we're looking at, so in these transcriptome studies that we talked about earlier, people have also raised the specter of, salamanders don't have, as many, they have a different repertoire of tumor suppressors and oncogenes and blah, blah, but, it's actually much more nuanced than that as far as gene expression goes. Like there are examples of mammalian tumor suppressors and oncogenes whose expression is turned on during regeneration. So it's not like, hit the gas, turn on the oncogenes and turn off the tumor suppressors. It's not really that simple, right? But we are thinking about this in terms of systemic activation, for example, because one of the things we want to know with systemic activation is if these cells elsewhere in the body are undergoing the first step and they're activating and re-entering the cell cycle and starting to proliferate, but then they're not going and making a new limb. Well, what happens in between there? How do you turn that response off? How do you extinguish it? Because also the animals aren't getting bigger everywhere. And what's the role of these like cell cycle regulators and tumor suppressor proteins, for example, in that process of extinguishing it? And, you know, how is that connected to their regeneration? I think that's really cool to think about, and probably also relates to some old papers that hinted that if a salamander has a tumor, you know, like on its tail, and then you amputate its limb, that sometimes it can regress at a distant location, right? Again, I would like to see those things revisited and, you know, more substantially, but I think they could be connected. And it's very interesting to think about. I hope more cancer biologists sort of try to look into salamanders as a source for ways to control tumor cells.
Me too. Me too. Jessica, tell us a little bit about what's happening with Matis Biosciences, because I mentioned at the beginning, scar products is going to be one of the focuses. And I think while we've been talking about regeneration, the other thing that these amphibians are really good at doing is not scarring and turning off of those processes. And obviously this yields not just potential, as you're working on for skincare, but also all sorts of runaway fibrotic processes and so forth that are behind a lot of the chronic degenerative diseases. Say a few words about what you're doing on this, and everything else you're researching. You're an entrepreneur as well, so please.
Yeah. I mean, there's not that much that I can really say right now about it. We call it Matisse.
Oh, sorry, Matisse.
Sort of one of my co-founders invented that name. It's sort of like a play on words with Matisse, the artist, because, you know, ultimately, you know, we want to restore the human body. It's, you know, a regenerative medicine company, a regenerative biology company, but we have made a very strategic decision to first go after indications in the skin, and that's for various reasons. But going back to how we got interested in this is that in the process of regenerating a limb, you can't even tell when it's done which limb was amputated. It's that perfect, right? They don't scar. And so we really want to know, like, how is regeneration connected to this anti-scarring? And there were a couple times in the lab for research on limb regeneration where we bumped into this topic. One of them is that repeated amputation study that we did. When I told you that if you amputate limbs multiple times, they stop regenerating, they don't get a blastema. And so we think they're failing in the early initiation stages. But if you look inside the limbs, they actually look a lot like human scar tissue. Very much like keloid scars, which are one of the worst kinds of scars because those, you know, are sort of exacerbated with time, and people who get them, you know, after almost any incision, and we don't really know like why some people are prone to them and others aren't. It's actually a really difficult topic in medicine, and so. What we know is that salamanders have some way of avoiding scarring during the normal process of regeneration. And so the premise of the company is basically to take the molecular insights from how super regenerators, and it's not just salamanders, but how super regenerators go about regenerating body parts, and to make the hypothesis that any of the things that they're using to regenerate body parts could also possibly have pro-regenerative and anti-scarring properties. When applied to mammalian or in fact human cells. And so what we like to think of is that human cells do have a latent ability, or human tissues have a latent ability to undergo scar-free healing. And we know this is true because as fetuses up to 24 weeks of gestation, you don't actually scar, right? It's just the process of maturing through development and then post-embryological development, something happens, right? And so we think that there's a latent ability for humans to be able to heal wounds scar-free. And one of the hypotheses we have is that if you provide them with the right instructions, which could be coming from insights in the salamander and other super regenerators, then we might sort of reawaken this. And so it's like a pretty basic hypothesis. And then what we're doing as a company is basically going in and asking, like, how, you know, which of the molecules could have these effects in human cells. and then ultimately taking them to people, because there's a huge need for these kinds of things. And in the medical setting, of course, like chronic wounds, et cetera, just regular surgical incisions, et cetera. But even from sort of like a cosmetic standpoint, the company was really inspired by something that happened to one of my kids. So like I'd been working on salamander limb regeneration for many years, and I knew they didn't scar, but I didn't feel like super compelled to go and tackle those questions, or certainly not to form a company, until one day my kid was out riding his bike. I was not there, and he had a super serious accident. Luckily, there was no car involved, but there was a lot of blood and an ambulance trip and a hospital transfer. And my kid had like smashed his whole front of his face up. He had a bad concussion, and basically like his some of his face bones like came through right in the middle, and he had to have plastic surgery. And I said, I felt so hopeless, not only in that day and moment, but like for the next 6 months, because here I am watching my kid grow a scar, and he's like 8 years old, right? And all I can think of is like, what does this mean for the rest of, his life? But I really realized there's nothing that I could go to the store and just get to try and, improve the outcome. And we had the best pediatric plastic surgeon in Boston. She was awesome. And she was like, you should massage it and put some sunblock on it. And I was like, great, I'll do those things. But also, what else can I do? I sometimes like to tell this story because, and he's fine with me telling the story, because if you see him now, I did massage the crap out of that, that incision. And you can still see it a little bit, but it's certainly not the first thing you see when you meet my kid, who's now 13. But it was the motivation for getting in there and saying, hey, I don't know anything about how to have a startup company, but I am in this ecosystem here where there are a lot of startup companies, and there are paths to doing it out, right in front of our faces. So let's just make a go of it, So this has been like a side gig for a few years. So hopefully next year you'll be able to go buy something in a, vial or a tube, and, play with it yourself and see if it helps when you get a boo-boo.
Awesome. I appreciate that. I'm sorry that your son experienced that, but I appreciate the story and once again, connecting back to nature inspiring, well, not just research in this case, but entrepreneurship and translational opportunities. I think it's so exciting, and really it's fascinating, Jessica, hearing you talk about all these themes. I mean, as I said at the beginning, I said I personally have a passion for this area, but just hearing you talk about it, and once again using these new tools to, as you say, to interrogate and understand further about what's going on in 2022 and beyond, it's the right time for all this to be happening. So it's really exciting. Wishing you the best with all this, not just the research, but of course the company work. For everybody that is going to be listening to this really exciting episode on the podcasts or watching on the YouTube channel, again, you've been spending time with Dr. Jessica Whited, assistant professor, stem cell and regenerative biology at Harvard University, also co-founder, Mattis Biosciences. Jessica, I want to thank you for taking the time out of your, sorry, Mattis Biosciences. I'm screwing things up today.
We're still kind of in stealth mode anyway, so.
Anyway, I want to pronounce things right. But anyhow, thank you for taking the time out of your schedule to do this. It really is fascinating. I want to thank you for obviously everything you're doing there at the lab and making these translational discoveries happen. And as we like to say on this show, thanks for helping to create a better tomorrow for so many people via the work you're doing. It's just a really fascinating story. And I really thank you for taking the time to walk us through it all. Really, really amazing work.
Thank you so much for having me. And I'm glad that we waited until the summer to do it so I could talk a little more about the results. But thank you so much. And I look forward to hearing it and hearing some of your other podcasts as well in the near future. So thank you, Ira.
Great seeing you.